Yay, hello everybody. Thank you, Tristan, for organizing this. This is really fun. This is a great experience so far. And I'll tell you about some recent work in our lab dealing with environment and experiences and changes that we see in area CA2. And yes, I'll be talking about, let's see, is this gonna, there we go. Yeah, how environmental enrichment and social isolation, they modulate inhibitory transmission. So this is a story that just came out in the hippocampal journal, Hippocampus. And here we go. You already got a really great introduction about the brain and the hippocampus and how we need it to learn memories and things. I'll just give you a brief rehash. Our lab really looks at the neurophysiology, the cellular properties of the cells. So with a little bit more focus on that. Yes, CA2, it's in all animals, it's in primates and rodents, and even though the naked mole rat, as Tristan is going to share with us. One aspect of this tiny part of the hippocampus is it has a unique molecular identity. I like to share this slide because it shows lots of different receptors that do important things, like metabotropic receptors, adenosine receptors. These really, you activate them, they activate a downstream sort of fairly powerful effect on the cell physiology. As you see, many of these are like highly enriched in CA2, if not exclusively expressed in area CA2. So the cells in this region, they have a lot of surprises still to share with us. And also another aspect that's interesting is that the extracellular matrix in this area is also very unusual. It's very complex. Typically, the paraneuronal nets surround inhibitory neurons. As you see in CA2, they seem to surround everything and everybody. Exactly what this is doing in this region is interesting. Other things, Tom went through this way better than I could, right? About all these sharp wave ripples and all the things we've learned recently about how CA2 is contributing to spatial encoding, sharp wave ripple generation, social recognition memory. And so this area is small. It seems to be playing an important role. And with this was also mentioned earlier, it's established for a long time, by Serena Dudek's lab, that when you stimulate these Schaefer collateral inputs and do either whole cell recording or field recording, you don't see the traditional long-term potentiation that you see in area CA1, right? So CA2 has this unusual property, right? And this, sort of puzzle, right? You have this strange area that's doing all these important things that doesn't behave like all the other pyramidal cells do in the hippocampus. Sort of motivated Vivian Chevalier a bit to look closer in this during his postdoc with Steve Siegelbaum. And here we're looking at some textbook recordings, right? Here's CA1, when you record, you get the postsynaptic potential. If you deliver a train of stimulation to CA3 inputs here, you can actually evoke action potential firing, right? And when you think of inhibitory transmission, and it's sort of like a very, very simple way, you can think about like a leaky bucket. You have all these little chloride ions in this sort of situation that are open and they're preventing synaptic inputs from depolarizing the soma. Let's say you block all the GABAergic ion channels and you block the leaky bucket. You see when you stimulate CA3 inputs and you record from CA1, that you get an increase in the postsynaptic potential, right? So far. Well, if you look in CA2, we have something very, very different. So you see, here's the postsynaptic potential. You have this tiny depolarization and this big hyperpolarization. And if you deliver a train of action potentials, you actually get a net hyperpolarization of the membrane potential, right? Which is really funky. And if you block a GABAergic transmission, wow, look, it looks like it can happen. CA3 can excite CA2. It's just, really, really, really prevented from this strong feed-forward inhibition. And so we thought, starting our team a long time ago now in Paris, okay, hey, let's study this. Maybe this is playing an important role in what's going on. We found that if you deliver a high frequency tetanus to CA3 inputs and you record here from CA2, you can induce a long-term depression in the inhibitory transmission. And you can deliver different kinds of stimulus to induce this long-term potentiation, this long-term depression, sorry. And we figured out that this LTD of inhibitory transmission is entirely due to the activation, of opioid receptors, right? So there's enkephalins getting released when you excite these CA3 inputs, activating delta opioid receptors presynaptically and preventing GABA transmission. We then determined, not going into details about this, that this plasticity is entirely due to the parvalbumin interneuron network in area CA2. So if you look at the parvalbumin cells in CA1 don't have the same properties. And if you excite them with 100 hertz, or if you directly excite the delta opioid receptors. And so this unique property, we were like, perhaps this is how CA2 is really contributing. Its role in the hippocampus. It is plastic. You have this way of modulating, of recruiting this area. And we tested this a bit further. So here's sort of a consequence we were excited about, that if you just silence CA2, and then you can either excite, or then you can either activate, or you just leave this cell, the PV interneuron network alone, or okay, so let me go through here again, sorry. What we do, we're inducing with a high frequency stimulation, this delta opioid plasticity, right, to get a long-term depression of the transmission right here. At the same time, we have AP5 to prevent traditional LTP everywhere else in the slice, right? So here, when we're going to record from CA1, and what we see is that if we silence CA2, right, there's nothing really changes, right? And the postsynaptic potential for CA1. However, if we leave things intact, right, we allow CA2 to do its thing, we see that inducing this long-term depression that's mediated entirely by the interneurons, you can actually increase the excitatory field potential in CA2 by stimulating CA3. So sort of this recruitment of CA2 can have some large consequences in other parts of the hippocampus. And if we take this right a bit further, so here we have this inhibitory network in CA2 that has consequences on CA1, assumably also from what Tom showed to CA3 as well. Another sort of changing directions here quickly, but it's really relevant that, well, CA2 has been unstudied for a very long time. Quite a bit was known. It was noticed in psychiatric disorders. So here's two examples. One is from this large consortium study where they looked at 224 different immunohistochemical markers in the human hippocampus from postmortem patients. And they found 3 that were significantly altered. And 2 of these, the parvalbumin total level on CA2 and the density of parvalbumin expressing cells in CA2. So here are these cells that we saw. Okay, these are really plastic. The transmission's plastic. They're really loaded in CA2. They're altered in postmortem schizophrenic brains. This was also noticed in a completely separate set of work by Francine Bennis, that postmortem studies that you have the density of interneurons in postmortem patients are reduced. So what we did a long time ago, now it feels like, is that we were able to use a mouse line, this 22q11.2 deletion syndrome line. It's the DeGeorge syndrome. And this chromosomal deletion, it's spontaneous. It confers about a 30% risk of schizophrenic diagnosis. So it's one of the leading causes of schizophrenia overall in the population. And we were able to collaborate with the Gogos lab, where they had made a transgenic mouse model with an equivalent chromosomal deletion on the chromosome 16. And it was previously described that these mice had many impairments that were sort of consistent with the DeGeorge syndrome in human patients. They had motor deficits, contextual fear conditioning, cognitive deficits, including delayed, non-matched to place. And importantly, they had this social memory deficit that really mirrored in many ways the social memory deficit to lesion mouse, like Tom had showed you, that Fred Heady had shown in the Siegelbaum lab. So here you see that you take a mouse and you introduce them to a novel mouse. for both the wild type and the deletion, you have fairly normal social interaction for the two normal mice. So they're social, right? They're not fearful of other animals. However, if you take a deletion animal and you introduce him again to the same animal, he continues to behave as if the familiar animal is novel. Whereas the control animal has he interacts less with the second presentation, right? So this is our readout of social recognition memory. And in our team, what we did, we performed the wholesale recordings to get a better understanding, okay, what's happening in area CA2 in these mice. And we saw that an age-dependent change in their intrinsic properties. And a really important intrinsic properties is the resting membrane potential, right? So we saw that as the animals, sort of emerged from adolescence, became early young adults, that we saw a hyperpolarization of the resting membrane potential. And a very natural consequence of this hyperpolarization is that it's harder for the cells to reach action potential. And so, and synaptic inputs come in, they don't reach threshold, and you have a very, very silent area CA2, right? So it's not surprising then that the animals behave like a CA2 lesion. And one thing that we noticed, which was kind of fun, is that you have this enrichment of this the RNA for a 2-pore leak channel, TREC1. This channel can be blocked by fluoxetine very quickly, which is an SSRI. This is Valium, so it's a Prozac. But it also is an ion channel blocker used like this. And this ion channel, it's very active in controlling the resting membrane potentials in many, many, many cells. And here in this series of experiments, I just showed the fluoxetine sensitive current when applying a ramp of, voltage clamp, applying a different ramp of potentials. And you see that the deletion animals, they have a much, much larger fluoxetine sensitive current. So they're much higher expression of TREC1. Other things we saw that were interesting, and here we're coming back to the parvalbumin interneurons, is that when we looked at the overall density of parvalbumin staining, we found in adult mice that there's a decrease specifically in CA2, in the stratum pyramidale and oriens, that we didn't see in CA1 and CA3. And this is very similar to what was reported in human postmortem patients. We did not see this in young animals. So this is indicative of this sort of age dependence to the disease onset in the animals in this model. And as you'd expect, right, you see fewer staining for interneurons. We also saw a reduced inhibitory transmission when we looked at it. So here's in control mice, you sort of see before and after blocking GABAergic transmission, the change in the potential that we can measure. This is really different in the deletion mice. And you see this over a range. Oh, again, this was also age dependent. So, okay. Right. So here's a summary so far, right? Here's our fun little circuit. We have CA2 right in the middle. And right, you have the CA3 inputs coming in. The dogma in the field that Serena's shown that, this input here is not very plastic at all. However, we do have this PV input, these parvalbumin cells. This is highly plastic, mediated by delta opioid receptors. And in this deletion animals that we're looking at, we have the loss of the staining of these cells. I can't tell you if the cells are gone. We can't detect them anymore. And there is a loss of GABAergic transmission, right? And around this time, right, I don't know, this paper was great by Pico Coroni, right, where he showed, or his lab showed, that you have these parvalbumin expressing network activity can really be influenced readily by their environment, by experience. And he took animals and it's stained, immunohistochemistry staining, looking at the parvalbumin concentration in control mice and environmentally enriched mice, or contextual fear conditioned mice. So here you have like very different experiences. And you could really see this change in the parvalbumin interneuron expression. And we thought, wow, okay, parvalbumin cells, they seem to be really important in CA2. What happens if we put if we put our mice and their cells, of course, they go together, we put them in EE for 3 weeks, what happens? And so that's what we did. Pretty simple experiment, right? We had our normal standard housing, 5 mice in a cage. And versus the large, we have very large cages. They have lots of toys, tubes and tunnels, and they have a wheel. They have little treats they like, little bacon flavored treats or fruity treats and shavings. This is like sort of fun for physiologists. It's like really fun. And so they live like this for 3 weeks. And then we looked to see, okay, what happened? So the first thing we did, we looked at the field potential. And we were surprised. We were very, very surprised. So this story took us a long time to figure out. So here we see it's just the local field potential. We do a high frequency stimulation, and as you can see, the SE, the control mice, right? Because you have an LTD of inhibitory transmission, you see an increase in the overall field potential, right? So this is a disinhibitory increase in the field potential. So this is reflective of the DOR plasticity I was talking about earlier. After enrichment, the plasticity we were very obsessed with, we're like, oh, this is the answer to everything. It really almost went away. It's really, really reduced, right? We're like, this makes very little sense. How can we explain this? So we tried harder. We did things differently. You know, we recorded whole cell. Okay, record from the cells. We'll look directly at the transmission, look directly at GABAergic transmission. And again, you see high frequency stimulation. The plasticity is really, really reduced. And compared to standard housing. So it's okay, how can you have this increased environment, this enhanced environment really seems to reduce the plasticity. What is this? If you directly activate the delta opioid receptors, again, the plasticity is nearly gone. So here we see this is this PV interneuron network that we were studying. Again, yep, the plasticity has changed. And looking at the field potentials also. So this is going the wrong place. So here we see that, no matter how we measured it, there was a big reduction in the plasticity of the PV plasticity. And as we saw too, we could measure, this takes about 2 weeks before it's really stable. We stayed with the 3-week paradigm because that matches with the literature. And but we saw that this does take some time to set in. And so the question is, okay, what's going on? What could be happening, right? Are we completely wrong? Is this plasticity not actually important in the hippocampus? And one hypothesis that we had was perhaps the experience of being in the enriched environment is actually inducing the plasticity at the PV-CA2 network, right? And so how would you test this? What would you expect that if this in the long-term depression is already induced, then we'd expect a reduction in GABAergic transmission. And one way of doing that is that we'd previously found on the parvalbumin network in CA2, if you inhibit a receptor called ERBB4, that results in reduced GABAergic transmission. So here, this is just an example from a whole cell recording. You add this inhibitor of this receptor and GABAergic transmission reduces. So we tried this in EE animals, right? Completely independent of the DOR plasticity. But we know it's in the same population of interneurons. And we saw that in the standard control caged animals, we saw the decrease in GABAergic transmission, whereas in the environmental enriched animals, not much change, right? Indicating that the GABAergic signaling has been altered for sure. And we're interpreting this as like, okay, GABAergic signaling is already low. So, inhibiting the ERBB4 receptor is not going to have much of an effect. And we saw that in the whole cell. We also saw it in the field recordings. And another prediction you could make if GABAergic transmission is reduced is that you can look at the spontaneous inhibitory postsynaptic current frequency. And what we found was that compared to standard housing, the environmentally enriched mice, that the frequency of these spontaneous events was decreased, while the overall amplitude was unchanged. Again, this is consistent with reduced GABAergic transmission. And again, the textbook sort of analysis of the paired pulse ratio, right? If you give 2 pulses and you look at the same synapses, take the ratio of transmission from before and after. We compared all of our experiments sort of before and after we induced the plasticity. for standard caged mice and for EE caged mice. And we see, if we just look at the averages, which is a bit cleaner here, that the paired pulse ratio with EE starts out appreciably higher before we do any plasticity induction. So this is consistent with the idea that the GABAergic transmission is reduced following enrichment, right? So we're like, okay, interesting. Let's see. We have this 22Q model mouse, right? What happens if we put it in enrichment and an enriched environment? Does the social memory improve? So here we do a habituation experiment where we take two mice, we expose them to each other multiple times, and then they get exposed to two new mice. We see in the wild type controls, environmental enrichment doesn't really change their social memory, which isn't a surprise, right? However, in our deletion mice, you see that when they're in standard housing, there's no significant difference between the investigation time here. However, in EE, there is a very significant difference. So the mice, they no longer behave as if it's a novel mouse. They sort of reduce, they habituate with this, just the change in caging conditions, nothing else. So then we're like, okay, let's see what's going on, what's changed, right? If we really think about the parvalbumin cells, when we're able to actually get rid of the delta opioid receptor, we see a kind of a mild, sort of the parvalbumin cell plasticity. If we really prevent it, it prevents habituation on the first and the second trial, but then eventually the mice do habituate. They do display social memory. So we're wondering, okay, if you have occlusion of this, how can you get such a big improvement in the deletion mice? And there's a part of the story that I have to explain because this took us a while to figure out. And so you have these parvalbumin cells, and here we're recording field potentials and just normal mice, right? And we record the field potentials, we give a high frequency stimulation. And then after this stabilizes, we give a second tetanus, a second stimulation here to CA3. This one's at 10 hertz, right? And we see that we were able to induce a second form of, we saw the second increase in the field potential. And then we're like, wow, what could this be, right? Here, if we continue looking at the fields, right? So here is like all the data put together. We've already induced a high frequency tetanus. So the DOR plasticity at the PV cells, this is already induced. And here we see this, this increase in field potential. Here we do a separate experiment where we add the control, we do nothing, right? And here we add DP-DPE to induce the ILTD, here, this PV CA2 synapse. And after adding DP-DPE, so we see once we induce the plasticity here, we're able to get a second plasticity, right? And we saw that, I see my cartoon has changed. This second plasticity can be blocked with the endocannabinoid receptor blocker AM251. It's also not present when you completely block GABAergic transmission. So this is a second form of disinhibitory long-term depression or disinhibitory increase in the field potential in area CA2. And so here we have this parvalbumin, delta opioid receptor plasticity. And then this will enable a second form of plasticity through the CCK interneurons, through the endocannabinoid signaling. We found that if this plasticity can be induced by firing action potentials in the CA2 pyramidal cells. So here there's no stimulation, where here the stimulation is only evoking the synaptic events. Here, what's happening is that the cells are getting current clamped very briefly and currents injected to allow action potential firing. And then following that, you can see a very large increase in the postsynaptic potential. This increase is blocked by ECB, like endocannabinoid receptor blocker, as well as GABAergic blockers, right? And so here's just a little summary so far. So we have a delta opioid receptor plasticity at the PVCA2 synapses. This allows action potential firing. Once you have this, activity from CA3 can drive action potentials in CA2. If you have action potential firing, then that results in endocannabinoid release. And so then you get a second plasticity here at the CCK synapses. So you have these two plasticities that are linked, playing a role, really enabling, controlling the activity of CA2. Time. And so I'm not going to have time to go through the details, how this is figured out. But this is really, Vivian Chevalier had already demonstrated during his postdoc that endocannabinoid signaling in CA1 is a very different monster. It's a very different beast. You can activate it with metabotropic glutamate receptors. It's very localized. You can have a whole series of inputs that undergo plasticity, but it's not really dependent on the cell. In CA2, it's a very different dynamic here because it's really linked to the cell itself firing. All the inputs coming onto CA2, coming onto that particular cell, will have given off the 2AG and sort of cause the LTD at the incoming inputs. So it's sort of interesting importance for the potential for encoding if you have cell-to-cell separate plasticity. So in collaboration with Laure Verre at Toulouse, we were able to do some behavioral testing of this ECB-mediated plasticity, where she was able to infuse either the blocker, AM251, or a vehicle, while the animal went through this social dishabituation task. And we saw that blocking the endocannabinoid receptors was really, really, really effective at preventing habituation. This was quite exciting to us. It seemed much more effective than the delta opioid receptor inhibition by far. And so just again, so if you lose the PV transmission and the 22q11 mice, right? So here we're back at the model mice. So we have a loss of PV transmission. We see that the cells are hyperpolarized. One question we asked is, okay, if we have this model mouse, are they able to undergo this ECB plasticity, right? So here we have the wild type. Now that the 22q it's called large deletion. And if we pre-activate all the PV DOR with incubation and the DOR agonist, we see the wild type animals undergo the ECB plasticity, no problem, but the deletion mice do not at all. And so we thought, maybe this is what's really explaining the social memory deficit in this line. Perhaps this is what's happening. They're hyperpolarized, things are, their inhibitory network's messed up. This is not functional. We find if you actually inject current, you can force the cells to fire, you can still induce the plasticity. So they have what it takes, they could, but you just have to get them to fire action potentials. And if we do that a bit artificially with fluoxetine, so we block TRK channels in the mutant mice, add, with fluoxetine, you can actually induce, we activate the PV network, we can induce the ECB plasticity. So it would be possible. And perhaps, right, maybe this is how the rescue is happening in the environmental enrichment. So perhaps, it's like these changing, you occlude, you reduce this plasticity enough, CA2 can start working again. And one thing I haven't told you about that we're also trying to figure out in parallel was that, yay, when you do whole cell recording from cells that were in enriched environment, you see a change in the intrinsic properties, and in particular, the resting membrane potential. So here's just the wild type mice. The cells become more depolarized, right? The membrane resistance also becomes a bit higher. The AP threshold is lower. And this delta here is the difference between the membrane potential and the AP threshold. It's substantially changed, right? So if we look at the deletion animals, that their cognition can be rescued, what we see is also a change in the resting membrane potential, the RM and the AP threshold. So we think, perhaps it's all coming together. Perhaps it's finally making sense, right? So with EE, we can record from these deletion mice, you can stimulate synaptic input, and you can actually get AP firing from the changes in the intrinsic properties. And this is evidence from, of course, input-output like this, but if we look at the population spike, so this is just a field recording looking at the group activity of different cells, either in the control animals or In the deletion animals, we see that spending 3 weeks in an EE condition, you can actually have evidence of action potential firing with synaptic input stimulation. And the thing that working out all these changes in the intrinsic properties, we come back to the TREC channel. A lot of this, we believe, there's lots of really strong evidence that these changes in intrinsic properties with EE are due to a reduction of the TREC current. So here we see that between SE and EE, if you block the TREC channels, everything depolarizes to about the same place. And this is something that the Donegan et al. In the Siegelbaum lab had already shown that you could rescue social memory by blocking TREC channels. So it all sort of comes together. Here we're able to have the same thing by just enriching the environment. And it seems to follow the same molecular mechanism. And just showing again more of the evidence of the fluoxetine, you see that the membrane potential becomes more depolarized, all reaching about the same place for the deletion in the wild type animals. And just another rehash again. So you have this ILTD is occluded. We have less inhibition here. VREST becomes more depolarized in both the wild type and mutant mice. These guys can fire action potentials. Then with interacting with a novel mouse will induce this ECB plasticity, allowing them to encode and learn. And you have nice normal behavior, right? So that's the prediction, right? Does it actually happen? Yay! So here we have wild type mice. Sorry, I can't help it. Anyway, so we have our wild type animals. And in this case, we've completely blocked all delta opioid receptor transmission, right? So if we look at these in SE, right, we do a 10 hertz stimulation, we see no plasticity, right? We take their littermates who've been living in EE conditions, and we give this tetanus, and we see, yay, we see a big increase in the field potential. This increase in field potential is blocked by the endocannabinoid blocker. So it seems that just having the mice in EE conditions for 3 weeks is really permissive for allowing the endocannabinoid plasticity to occur separately from the delta opioid receptor plasticity. And luckily, because we see this, our interpretation of the rescue, we can induce the ECB plasticity in the deletion mice after enrichment. So that's what we see here in this dark magenta trace. So these are deletion animals that have been living in an enriched environment. And you see if we block the endocannabinoid receptors or we keep them in standard environment, we don't see this plasticity. So what's the mechanism? Okay, TREC1, right? It's all about the leak channels. It's all about this stuff, but what's really regulating the channel? And we have some clues. We're working on it. We're really trying. We have a few clues. These channels are not friendly. It seems like you look at them funny and they change their conductance. They're really annoying. But we're now realized we've zoomed in that it's probably through this lipid signaling. So the lipid composition can really change the open probability of these channels. And we have a friend who's down in Nice, and she studies these inflammatory responses. And she's really interested in stress-induced inflammatory responses. And she wasn't looking for CA2, it just kind of jumped out at her, that if she puts mice for 3 weeks in an enriched environment, she sees a very specific increase in the secreted phospholipase in CA2 in the hippocampus. And she was really surprised. She sent me these images. She's like, oh my God, do you know about CA2? And I was like, yay, I think we found something. And using biochemical methods, she could detect different RNA expression of this secreted phospholipase. If you're like me and you have no idea what phospholipases do, I've learned since then, they cut off one of these legs of the lipids and really sort of change the membrane properties. And if you do a lipidomics analysis of animals that have spent time in an enriched environment, you see that in the hippocampus, or even specifically in area CA2, you have a very, very, very big change in many, many different kinds of lipids. And I don't really have the details of the experiments, but if you modify the concentration of SPLA2G5 and sort of biochemical tests, you can change the conductance of the TREC channel. And one thing we've done, that there's a blocker that exists for this. It's called LY33313. And if you put it in the water bottles of the mice, you can reverse the changes in the lipids. You can also prevent changes in the intrinsic properties of the cells. And Joelle has shown that if you block this, that you can actually prevent any sort of improvement in the social memory of the animals. And so we're kind of excited about this. We can effectively, make life worse for mice. Now, if we could somehow figure out, how to sort of hinge this mechanism to more therapeutic avenue, that's one thing we're interested in. And so here's the big conclusion slide. And one other thing, so what we see now, it's like CA2, it's easily modulated. You have this highly plastic parabolic network that gets influenced by experience. We see living in EE. There's data that I haven't had time to show you, but just a single social interaction will induce plasticity at this synapse here. This plasticity will then lead to AP firing by synaptic activity, right? Which can then lead to further plasticity at other interneurons. And this is very cell specific, so perhaps this plays an important role in encoding, we're not sure. And so this whole little circuit here seems to be really modulated by experience, by neuromodulators. And just one further example of that that I'll share with you quickly. And this is really an observation that the student made, was that the last mouse in the cage, right? So what she did, she would take a mouse out of the EE cage and do field recordings every day. And she noticed the mouse who spent 24 hours all alone, right? All the effects of enrichment were gone, right? Everything went back to looking like he'd spent 3 weeks in a standard environment. And so we're like, wow, it's a mild stress. It's not pleasant, but it seems to be fairly powerful for altering this inhibitory plasticity network in CA2. And so this is something else we're sort of curious about. Okay, what's the neuromodulator that's doing this? And we see that with the DPD, with the DOR plasticity, also with the ERBB4 receptor blocker. So this is CA2 has plenty of questions that are left. And again, if we take a zoom out, right, we have the hypothalamic inputs coming in, they target these PV cells. So this is interesting because this sort of network, consequences for a psychiatric disease and experience, the CA2 projects to CA1, but also back to CA3. So if you're modulating the overall activity and output, you can do big things to lots of places. Not even thinking about CA2's projecting to the septum. Wow, oh my God. All right, here we go. So anyway, and all the people who did the work, I want to really point out for the endocannabinoid story and the enrichment story of Vivian and Mete here, were big players. A lot of the behavior was done by Laure and Ludovine. And here's the current team. And please ask questions. Sorry if that was too fast, but that's, thank you. I have a little mouse at the end.